going to draw a very simplified phylogeny to kind of give you the idea of where the primary and secondary endosymbiosis events are happening in the context of the history of life. Usually I have you write some sort of short essay about this on the exam. All right, so start with your paper on your notes up and down, and you probably want some colors or at least some different kinds of writing utensils. Of course, you can always do this without color and do other symbols if you want to. Looking at primary and secondary endosymbiosis. And like I said, this is a simplified version. Whenever I am talking about primary, I'm going to use this purple color, and then I will use this yellow to indicate secondary events. One room here, then we're going to have another line there. These two don't have too much detail with them, so they can be kind of close together. I'm going to go to the bottom, and I'm going to branch off one at the bottom. And you want to leave some room on the right because you're going to have some arrows. I also am lining up all these modern day groups, like the tips of my branches, because they're all living today, so there's no extinction events and whatever. So, and like the branching on these, where this branch falls has no relevance to real time. It's just the pattern of branching that you want to look at here. Okay, and then we have this group here, which is going to split into three. So we've got, this is the sarclade, the A part of it, an example. So like I said too, this is simplified. So basically these are just examples within the groups. Draw the root of the tree there. Okay. And I'm doing a polytomy, as you can tell, because they're all coming off the same point here, which is because it doesn't matter about the branching otherwise. This is all in eukarya. So the first event is the primary endosymbiosis that leads to the formation of the mitochondria. So all eukaryotes have mitochondria that all appear to be from the same endosymbiosis event. Some of them have reduced mitochondria, slightly modified, or have mostly lost them, but they all come from the cell that underwent this primary endosymbiosis. So this is going to be a primary. I use I'm writing sideways, so this is a one with a sort of a not symbol to it. It doesn't perfectly mean primary, but it's a good abbreviation for me to make it fit, and so I can write it faster in my notes if I was taking notes in class, something like that. So this is a primary endosymbiosis of an alpha proteobacterium. So this is the Greek symbol for alpha, like a little fish. Proteobacterium. Or bacterium is singular, bacteria is plural. And I will put this in orange. And remember that alpha proteobacterium getting engulfed is not unusual at all. That's how lots of single-celled organisms eat. But the unusual part is that it didn't get digested, exchanged genes with the host cell. They became dependent on each other, advantage because now it could make, the host cell could make more ATP. And so that alpha proteobacterium turned into the mitochondrion. You'll also notice here mitochondrion is singular, mitochondria is plural. And then this is all primary endosymbiosis event. Everything comes from this. Then in one lineage, and we'll go down to the bottom now, there was another primary endosymbiosis event. This is where people get mixed up because they're like, oh, it's the second thing that happened. It's secondary endosymbiosis. But no, anytime you have a bacteria involved, so if there's some sort of bacterium being engulfed, That's always primary. Okay, so here's the other primary endosymbiosis, or one of the other primary endosymbiosis events. I'll just write endosymbiosis of what in this case? Cyanobacterium. And that did not get digested and became chloroplast. The chloroplast brings with it the chlorophyll A. There are other pigments in various chloroplasts depending on the group, but all of them have this chlorophyll A from the cyanobacterium. So that is a primary event. Now, which lineage did this happen in? The Archaeplastida lineage. So all this group here, this bottom group is the Archaeplastida. They're the ones that derive directly from the primary endosymbiosis of the cyanobacterium. They also have the mitochondria in them as well. And I'm going to be using a different green. This is sort of supposed to be a blue-green, or sometimes I draw blue and then like literally draw green with it. So what groups do we have in our Archaeplastida? So you do want to know this. One of them is an additional group that I include beyond the book because it's particularly important for evidence of this primary endosymbiosis event. It's called glaucophytes, the glaucophytes. Glaucophytes are pretty cool because their chloroplasts today actually have the peptidoglycan. So that bacterial cell wall material is in the chloroplast of the glaucophytes, which is a really beautiful piece of evidence of the fact that the glaucophyte chloroplast clearly comes from a bacterium. Then other groups here in the Archaeplastida. The red algae group, and then this third group up here, which is the green algae group, which is a polyphyletic group, and split into the land plants now. All the other chloroplasts, because you might be thinking, well, I know that, there's other algae, right? Like the diatoms and the dinoflagellates and that kind of stuff. Brown algae, where did they come from? They came from secondary events. Let's do the green algae. So in the history of time, a unicellular green alga being singular, was engulfed, I'll draw it at the top, by a already existing eukaryotic cell, single cellular, that was heterotrophic, did not do photosynthesis, and that would be normal because it would eat it, but then it didn't digest it and ended up becoming dependent on the host cell and vice versa. And we have the euglenids, which includes euglena. Their chloroplast comes from a green algae or a green alga, and it has multiple membranes around it. So that's one secondary endosymbiosis. I'll draw it like this for secondary. Remember, again, that's not quite meaning secondary, but it's close enough. There's multiple events here. Yellow is our secondary events. Another group that arose from engulfing a green alga is part of the Rhizarian clade. So this RH is the Rhizarian, and specifically this one's underneath the Cercozoa group there. Okay, it's the chlorarachniophytes. Arachnio, like spider. they're one of the types of the rosarians that have those filamentous pseudopodia. So that's where the name comes from. And the chloro means color. So they have color because they have chloroplasts, chlorarachniophytes. And phyte means plant-like, meaning that in this case it has chloroplasts and does photosynthesis. So that's where chlorarachniophytes comes from, even though it seems a crazy word. Okay, they come from the engulfing of a green alga. That is a secondary endosymbiotic event. And chlorarachniophytes are interesting because they have a very nice piece of evidence of secondary endosymbiosis. So I'm going to actually just, because I don't want to get too crowded, draw a little dot here. And I'm going to add this up to the top. There's so much evidence for endosymbiosis, but there's some unique groups. For example, let's add this in. The glaucophytes that we talked about before. Let's put a dot by them. And then you could write these things on another page if you want. Glaucophytes, their unique thing is that they have the peptidoglycan in their chloroplast walls, or sometimes they're called cyanel walls. That's peptidoglycan, remember, is the bacterial cell wall material. Okay. A unique evidence of the chlorarachniophytes, they have what is called a nucleomorph. All right, just briefly, the nucleomorph has, so it comes from this. So here's our original cyanobacteria. Now, the thylakoids are not separated in the cyanobacteria. They're basically attached to the walls, but it's a folded membrane and it has a cell or plasma membrane on the outside. So this is from the cyanobacteria. Then it gets engulfed and ends up being a green alga. So the engulfing event gives it a membrane from the plasma membrane, like it comes in a vesicle kind of thing. So this is the chloroplast, which ends up inside of the cell membrane of the green algae. And that means that in here, because it's a eukaryote, it has a nucleus. So this is the green alga. Then that gets engulfed, though, by another eukaryote, and this is the cell membrane from the secondary endosymbiosis, whereas this was the membrane from the primary endosymbiosis. And then the nucleus then becomes a nucleomorph. So basically, the chloroplast now isn't this little part of the inside. The chloroplast now is this whole deal here. Basically end up with a chloroplast with 1, 2, nucleomorph, 3, 4, and then the thylakoids inside of that, and that is the chloroplast. And there's your little nucleomorph. Basically, it's like a remnant nucleus. It does have functional genes, though, and is required for the chloroplast to be able to function. And then, of course, this is inside of the protist, the chlorarachniophyte in the case that we're talking about. The other groups that have chloroplasts from secondary endosymbiosis are not from the green algae chloroplast, they're from red algae chloroplast. So this happened, looks like a bunch of times. It's pretty straight. Okay, so one group that engulfed a red alga were the haptophyte group. Haptophyta, specifically coccolithophore. Okay, so coccolithophore comes from a red alga. This is the other group that actually has nucleomorph in it still, but it's from a red alga. This group is called the cryptomonads. Cryptomonads. Okay, so they're from a red alga, and they have the interesting nucleomorph as well. Those are the only two groups that are known, chlorarachniophytes and the cryptomonads. So let's give them a line. Okay. All right, so now the next group we have is the alveolate clade. This is a wild and crazy group, is what I'll call it. The dinoflagellates, many of them just come from directly from a red alga. Then we see that there's some other endosymbioses here. Not all dinoflagellates have chloroplasts. As far as I know right now, all dinoflagellates, even if they have chloroplasts, though, can also eat, seems to be. So the question is, did some of them gain, some lineages of dinoflagellates gain the red alga and then diverge? Did the original one have the red alga and then groups lost it? There are dinoflagellates that have colorless, like pigmentless, chloroplasty plastid type things, suggesting that it was a chloroplast and then it lost the pigment genes and such, and now it's non-functional. So dinoflagellates are pretty diverse in those, but for sure, many of the dinoflagellates have their chloroplast derived from a red alga. And it's possible that all of them have this originally, and then they may have lost it instead. So those are important group here. Then we've got our two groups under straminopiles. Diatoms and brown algae. Okay, diatoms, brown algae, and the haptophytes all have fucoxanthin as their pigment, in addition to chlorophyll, but fucoxanthin, that brown pigment. Dinoflagellates, most of them have their own unique pigment, so there's all kinds of changes happening. Another thing I want to point out is that there is at least one additional primary endosymbiosis. Okay, so that was all secondary. Now we're engulfing green algae. Let's go back to engulfing cyanobacteria for a moment. So this is another cercozoan group. The genus is Pollinella. That's why I'm underlining it, because it's actually the genus. And this group, actually, I'll draw it in blue, comes from engulfing a different cyanobacteria. And we call it a chromophore instead of a chloroplast. And so we're going to go back and mark this with primary endosymbiosis. Okay. And both of these groups are cercozoans within the Rosaria. So that is your primary and secondary endosymbiosis. Tertiary means third. So I'm not going to need you to know the super details about which groups do this and all. I mean, yeah, which groups derive from each of these, but I do want you to have a familiarity with it. So particularly, I mentioned the dinoflagellates are wild and crazy. This is why I say that. So what it looks like is that we have certain types of dinoflagellates, and they got their chloroplasts right from the red algae. It looks like that dinoflagellate then engulfed a haptophyte, which we also said got its chloroplast from red algae. Let's use some pink. Okay, so this is going to be like a tertiary event. And then it looks like it loses the dinoflagellate chloroplast. So then you end up getting a dinoflagellate species with a haptophyte like chloroplast. So basically these species don't have the normal dinoflagellate pigment in them. Instead, the pigment they have is, and other characteristics they have, look like what the haptophytes have. It looks like the same sort of thing happened with the dinoflagellates. And another group that got theirs from a different red algae, the diatoms. And you end up getting a dinoflagellate species with a diatom-like chloroplast. That would be due to a tertiary endosymbiosis event. There also appears to be some serial secondary endosymbiosis. So this again is in the dinoflagellates. And so you have a dinoflagellate, which again gets its chloroplast from the red algae. So it has that already. But then what you see inside of that dinoflagellate is actually a chloroplast that looks like a green algae chloroplast. So what seems to have happened is that you had a one-celled green alga, so from that Archaeplastida group, and the green alga got engulfed by the dinoflagellate, not digested. Red algae chloroplast loses functionality. And so then what you end up having in the end is a dinoflagellate with a green alga-like chloroplast. All right, so now you have an overall picture of the primary and secondary endosymbiosis events in the history of life.